Hello. Today is the seventh problem solving session for the course Environmental Chemistry and Microbiology. I am Shrija Ghosh, a PMRF from IIT Kanpur, and the course instructors are Professor Anjali Pal and Professor Sudha Goyal from IIT Kharagpur. So till now, we have been discussing about different topics related to environmental chemistry. From today onwards, we'll start a new topic, that is microbiology. So today we'll be so it will be like an introduction to microbiology. So we'll be discussing questions related to your assignment that is given for week 7, and we'll be discussing the questions related to the topic, that is introduction to microbiology, and uh, some of the microbiological techniques and terminologies that we use. Okay, so I'll start with the questions, and meanwhile, if you have any doubt in any of the question, you can ask me. So I'll start with the first question. Which of the following famous person was first to find out that microorganisms are responsible for wound infection and develop the method of antiseptic surgery? Which of the following famous person was first to find out that microorganisms are responsible for wound infection and develop the method of antiseptic surgery? Option A, Robert Hooke. Option B, Paul Ehrlich. Option C, Joseph Lister. And option D, Louis Pasteur. Okay, I have one answer, option A. Any other guesses? So, who among the following have developed the method of antiseptic surgery? If Smita is saying option A, that is Robert Hooke. Anyone else? So, we have Robert Hooke, Paul Ehrlich, Joseph Lister, and Louis Pasteur. So, the correct answer is Joseph Lister. So, Lister actually saw that a lot of times when the surgery was going on, during the surgery, the wound of the patient that was developing infection during the surgery or post surgery, and due to those infections, a lot of patients were dying. So, he tried to understand why this is happening, and he found out that okay, there are certain microorganisms that can uh, start causing infection in the wound. So nowadays we all know that whenever we get a cut, we apply Dettol or some antiseptic uh, liquid, right? Because we don't want any infection to develop in that area. But at that time, nobody knew that this was the case, and that is why uh, Lister was trying to find out a method so that wound can be sterilized properly,
పొద్దునే
and
కదరా గ్రూప్ లో పెట్టాను
it it won't develop a infection. So Joseph Lister actually developed this method of antiseptic surgery where he used carbolic acid to kill the germ. So he found a way to prevent infection in the wounds during and after surgery. He was the first to apply the science of germ theory to surgery. So germ theory of diseases postulated by Robert Koch, and uh, we'll discuss about that. So he basically had hypothesized or stated that uh, any microorganism that can cause a disease, uh, if we isolate that organism, and then infect a healthy individual, that healthy individual will also develop that disease. So basically, a disease is caused by a microorganism. That relationship between a pathogen and the disease, that was postulated by Robert Koch, and that theory was called germ theory of disease. So Joseph Lister actually applied that germ theory, that okay, a microorganism that can cause a disease, that he applied it to surgery, that during surgery in a wound certain infections are developing, and those infections are also kind of like a disease, right? So those infections that are developing, that are basically due to certain microorganisms, and to prevent that, we need to kill the microorganisms. Basically, we need to find out a method to sterilize the wound area, so that no organism will be able to grow. Kindly mute your mic. If you have a question, then you can ask. Uh, so, Lister's antisepsis system is the basis of modern infection control. So, nowadays we all are using antiseptic liquids or antiseptic lotions whenever we get a cut or wound. We apply those antiseptics to prevent infection. But in 1867, when people were dying of wound infections, when they did not know how to control that, Joseph Lister was the first one who uh, developed an antiseptic surgery procedure. Okay, so it's because of him that everyone, almost everybody, is aware of uh, the usage of antiseptic these days. Okay, and as an antiseptic, he used a carbolic acid solution. So he mixed carbolic acid with oil or water and then used it over the wound. So you can read about his story, how he discovered it. It's actually interesting. Okay, and the other options that we had, that is Robert Hooke, Paul Ehrlich, and Louis Pasteur. So Robert Hooke was the first one who observed a cell. So he was the first to report uh, about cell. So he was the first to observe a cell under a microscope. Louis Pasteur has contributed to science in many different ways. So he has developed it, like he has developed the fermentation, like found the reason of fermentation, that microorganisms are involved in fermentation. He has disproved the spontaneous generation theory. Then pasteurization is a sterilization technique. We all know the milk packets we get in market, they are pasteurized milk. So that is basically a ultra heat, uh, ultra high temperature sterilization method. So at a very high temperature, the milk is heated so that all the microorganisms are killed inside that, so that it is sterile, and it won't develop any uh, like it won't cause any infection or disease. Then Paul Ehrlich, he was the first uh, to give the effective treatment for syphilis. And in 1910, a drug for syphilis was released called Salvarsan, which was developed by Paul Ehrlich. Okay, that's clear to everybody. So this is basically history of microbiology, how microbiology developed over the years. So first time a person observed a cell, and then first time a person observed a bacteria under a microscope, and then how these microorganisms are associated with the disease, or how these microorganisms are associated with the fermentation process, which is a very age old process used in cooking, used in a lot of industrial processes, in alcoholic beverages, in your household uh, uh, food, in many of the Indian preparation, dosa and all those batters are fermented batters, and every fermentation process includes a microorganism. So Pasteur was the first one who determined that microorganisms are responsible for fermentation. And similarly, there are a lot of this development that had happened in uh, the field of microbiology that led to the current research that has been going on in this field. Because of these uh, people only, we are able to do the research in today's date and time. Okay, so the next question, moving on to the next question, question number 2. In which year was influenza virus first isolated from human sample? Anybody knows the answer or wants to answer? In which year was the influenza virus first isolated from human sample? The answer is 1933. So, influenza virus. So, you all are aware of the coronavirus pandemic that we all have gone through. So, similar to that, in early 1900s, there was another pandemic which was due to this influenza virus, I think in 1918 or something. And at that time also there was a lot of uh, uh, cases, and a lot of people were dying due to this viral infection, and it became very severe. And at that time there was no treatment on, even the causative agent was not known. At that time virus was not very thoroughly studied. So in 1933, Wilson Smith, C.H. Andrews, and P.P. Laidlaw, they isolated the human influenza virus. They were the first to isolate this virus. They were the first to tell the world that this disease was not due to a bacteria, but it was in fact due to a virus. They did not find any bacterial sample in the uh, saliva or sputum, but they found a viral particle. So they were the first to discover the human influenza virus, and because of their discovery, later all the vaccines related to influenza were developed. Okay, so because they could isolate the virus, the vaccine development was possible for the virus, because there was no antibody generation in the body by which they could develop the vaccine. Okay, so this was also a very important discovery in the field of microbiology. Moving on to the next question now, question number 3. Defining characteristics of living beings are: option A, they reproduce; option B, they move in response to external stimuli; option C, they can grow and repair their own tissues; and option D, all of the above. The defining characteristics of a living beings are: they reproduce, they move in response to external stimuli, they can grow and repair their own tissues, and D is all of the above. What do you think defines a living being? Okay, 2 people have answered option D. What about others? This is a simple question. You can read the options and uh, try to answer. What about others? You don't want to answer, or you don't know the answer? So option D is the correct answer. So reproduction, movement in response to stimuli, growth and repair of their own tissues are all the defining characteristics of living being. So a living being has multiple characteristics, like there are multiple criteria that can define if a particle is living or non-living. So first is metabolism. Cell is an open system that takes nutrients from the environment, transforms it into biomass and energy, and the output is waste excreted into the environment. So that is metabolism. We eat food and then we excrete it out. So that is how the cycle goes on. Okay. So if any cell or cellular system is able to uptake nutrients from the environment, transform it into energy, and then output as an output, it can excrete the waste out into the environment itself. That means that body is the living body. Then comes reproduction. If a cell is living, it should be able to reproduce themselves based on their genetic code. Okay, so cell reproduce themselves based on their genetic code. So if there's a living being, they will be able to reproduce. Their cells will be able to multiply in numbers, increase in number. Okay, sorry. Then comes differentiation. Differentiation means cells can adapt to new environmental conditions, such as changing into spores to survive in hostile conditions. So there are certain bacteria or certain organisms who can change into spores to survive harsh conditions, to survive hostile conditions where they know that they won't be able to survive as it is. They develop into some kind of dormant structure so that they can live through those conditions. So adaptability is a criteria for a living cell. If a cell is living, then they will be able to differentiate, they will be able to adapt to new environmental conditions. Then comes communication. All living cells are able to communicate in one way or the other. So we humans are communicating by talking to each other. Then there are certain animals who release certain kind of chemicals, or who have certain kind of sound response, or some kind of visual response. So every living organism can communicate with each other. So even a single celled organism, even a bacteria, a single cell organism, they can also communicate with each other by releasing certain chemicals and changing their surrounding environment, so that the cell decides, it can sense that and understand, okay, this is how this cell is responding, so I should respond this way. So that's the way every living organism communicate. Okay, so every living organism has a way of communication. It need not be like how we are communicating by talking to each other, but there is a way of communicating. So chemicals released or taken up by cells are used to signal changes in the environment, etc. Okay, then comes movement. If a cell is living, then it will be able to move. So cells can move in response to light, chemicals. So humans can walk, but plants can't walk. But even plants are moving. They move in response to sunlight, right? Same way, every organism, multicellular or unicellular, every organism is able to move in some way in response to some chemical or some light. Okay. Then comes evolution. Cells modify themselves to adapt to new conditions, resulting in new species over period of time, long periods of time. So cells are able to evolve. Okay, that's how the whole diversity has uh, been established. So these are the characteristics of a living cell. So if a cell if cell or cellular body will be defined as living, these characteristics should be present in that body. Okay, I'm turning off my video to save the bandwidth. It's lagging a bit. So moving on to the next question now, question number 4. Who explained the cause and effect relationship between a pathogen and a disease? Who explained the cause and effect relationship between a pathogen and a disease? Option A, Edward Jenner. Option B, Vinogradsky. Option C, Van Leeuwenhoek. And option D, Robert Koch. I have already told you about this in the first question, so you should be able to answer that if you remember. So, who explained the cause and effect relationship of a pathogen and a disease? Edward Jenner, Vinogradsky, Van Leeuwenhoek, or Robert Koch? I have one answer, option A. What about others? Anyone else wants to answer? At least try. Okay. One more person has answered A. So the correct answer is actually option D, Robert Koch. So Robert Koch actually gave the germ theory of disease. I mentioned this in the previous uh, slide. Uh, so basically, germ theory of disease explained why a disease is like explained that a disease is all caused by a pathogen. So the relationship between a pathogen and a disease that was given by Robert Koch, and he uh, gave four postulates uh, that is known as Koch postulates. So Koch's four postulates. So they are: first is microorganisms must be found in abundance in all organisms suffering from a disease but should not be found in healthy organisms. So basically, if I am suffering from a disease, if I am suffering from a disease, there must be a microorganism present inside my body, which should not be present in your body if you are the healthy individual. Okay. So if I am the diseased one and you are the healthy individual, there must be some microorganism that is present in my body that is causing the disease, which is not absent in your body. Okay. Then the second postulate is microorganisms must be isolated from a diseased organism grown in pure culture. So that microorganism should be uh, isolated, like it should be able to, like anybody should be able to isolate the microorganism that is causing the disease. So if I am the diseased individual, you should be able to isolate the microorganism that is causing the disease in a pure culture. You should be able to grow it in a pure culture. And then the third postulate says the cultured microorganism should cause disease when introduced into a healthy organism. So if I have a disease and you have isolated the microorganism that is causing the disease and grown it in a pure culture, now if you inoculate that, if you introduce that microorganism in your body, there's a chance that, so according to Koch's postulate, you should also develop the same disease. Okay. And then fourth postulate is microorganism must be re-isolated from the inoculated diseased experimental host and identified as being to the original specific causative agent. So basically, after you have also developed the disease, then again from your body that microorganism can should be re-isolated, and then it should match with the microorganism that was taken out from the diseased individual originally. Okay. So if I'm the diseased individual and you isolated a microorganism from my body, introduced it in a healthy individual, after that, that healthy individual should also develop a disease. Now, if you again isolate the microorganism from that diseased individual and you match it with the microorganism you had previously isolated from my body, they should match. Okay, so that was Koch's postulate. Basically, he defined the relationship between the pathogen and the disease. So, causative pathogen and the disease. So, disease and the pathogen causing that disease. Okay, so he defined the relationship between that. The other options were Edward Jenner, Vinogradsky, and Van Leeuwenhoek. So, Edward Jenner was actually the first person to develop a vaccine. So, he developed a smallpox vaccine. So, what he did was he was actually trying to find out a way to prevent smallpox. At that time, smallpox was widely spread. And what he did was, so there's another related uh, disease called cowpox. So cowpox was also caused by a microorganism. So what he did was he took a small sample of the pus from an individual suffering from cowpox. Okay, and introduced it into the individual suffering from smallpox. So, the person, when introduced with the pus of the cowpox, uh, did not develop smallpox. Okay, so that's how he understood that if we introduce an attenuated virus inside a body, the body will kind of identify that, and body will recognize it as a diseased particle, and it won't let the body to develop a disease when actual virus will attack. Okay, so that's what Edward Jenner did. Edward Jenner developed the smallpox vaccine. Then Vinogradsky, he actually pioneered the cycle of life concept. He had developed a Vinogradsky column. It's a very classical science experiment that is done in a microbiology lab. So we take a large column or a large measuring cylinder you can take and fill it with pond water and mud and set up a mini ecosystem, and then add some newspaper cutouts and eggshells as a carbon source, as a sulfur source, and then cover it from top with few holes in it, and then keep it in the sunlight. So what will happen? You will see a whole ecosystem starts growing in that column, in that measuring cylinder, after a month or so, and you will see there's a division of the organisms. So at the bottommost layer uh, you will have the anaerobic cells, and then in the middle layer you will have the microaerophilic cells, and like that. So I'll just use my pen. Just give me a second. It would be easier to explain. Yeah. So it's Vinogradsky column is basically like a long measuring cylinder. Okay. So this measuring cylinder, you fill it with pond water and mud, and so mud will settle down, and you can add newspaper cutouts and eggshells and egg yolk as a carbon source, as a protein, as a sulfur source, basically. and then leave it out in the sunlight. And then slowly a lot of microorganisms will start growing, and they will divide themselves in the column, such that anaerobic bacteria, so green and purple sulfur bacteria, will be at the lower region. So green sulfur bacteria and purple sulfur bacteria. Then you will have some microaerophilic bacteria at the, like, just below the surface, and at the surface you will have the cyanobacteria that are aerophilic and that are photosynthetic organisms. So like that you will have a division. So here the oxygen level will be high, so all the aerobic bacteria will grow. Here the oxygen level will be medium, so here the oxygen level will be medium, so different other organisms will grow. Then here the oxygen level will be very low, so anaerobic growth will be there. So like that, so Vinogradsky developed a column to describe the cycle of life. Okay, and then comes Van Leeuwenhoek. He is known as the father of microbiology. He was the first to observe a bacteria under a microscope. So he had developed a very basic microscope, and he had first identified a bacteria and a protist cell. So he's he was the first to start this microbiology journey. Okay. So these are some major milestones that this is also mentioned in your lecture video. So I've taken it from there only. So I just added it in the slide because we are talking about so many people. You should know the contribution to the field of microbiology, and more or less the years. Not all the years can be like, no one can memorize all the years, but at least you should know the name and their discovery. So Robert Hooke we had discussed, Leeuwenhoek. Then Carl Linus, he has given the nomenclature of organism. Then Edward Jenner we discussed, Louis Pasteur we have discussed, Lister, Koch, Vinogradsky. Then comes Fleming, Chain, and Florey. They have discovered penicillin. Then Beadle and Tatum, they have given the relation between the genes and enzyme. Then Avery, MacLeod, McCarthy, they have a very nice experiment to describe the genetic material of all organisms as DNA. Then Watson and Crick obviously gave the structure of DNA. So these are just some of the major milestones in microbiology. Okay. So moving on, next question is, prokaryotes can be found in the following two domains. Option A, bacteria and protista. Option B, archaea and eukarya. Option C, bacteria and archaea. And option D, eukarya and monera. Prokaryotes can be found in the following two domains. Which of the following two domains do you think prokaryotes can be found? Bacteria and protista, archaea and eukarya, bacteria and archaea, or eukarya and monera? Come on, what do you think? Okay, Ipsmita says option C. What about others? Try to answer it, otherwise, how will I know if you are understanding, if you know the answer? If you have any form of doubt, you can ask. Anyone else wants to answer? The correct answer is option C, bacteria and archaea. So you are right. So living organisms are divided into two categories mainly, prokaryotes and eukaryotes. Prokaryotes are unicellular organisms. Under eukaryotes, you can have unicellular or multicellular organisms both. In prokaryotes, uh, archaea and bacteria, eubacteria or bacteria, and archaebacteria or archaea. These are the two classes that are there under prokaryotes. Under eukaryotes, I told you there are either unicellular or multicellular. So under unicellular, you have algae, protozoa, and fungi. Among fungi, there are some fungi who are which are unicellular, and there are some which are multicellular. So yeast is an example of a fungi that is unicellular. Then under multicellular, also you have fungi. So there are other fungus which are multicellular. Even the mushroom that we eat, that is also a fungus, and that is a multicellular fungus. Then plantae and animalia, that is the plants and the animals. So we come under animalia. So this is how living organisms are divided into two classes, prokaryotes and eukaryotes. So this division, prokaryotes and eukaryotes, it is based on certain differences. There are principal differences between the prokaryotic cell and eukaryotic cell. So first comes the size. The size of prokaryote is very small as compared to eukaryote. You can see 0.2 to 2 micron is the size for prokaryote, whereas 10 to 100 micron is the size of a eukaryote. Then the major difference between a prokaryote and a eukaryote is the nucleus. So in prokaryotes, there is no nuclear membrane present. So the genetic nucleus is the place where the genetic material is present. Okay. So for prokaryotes, the genetic material, that is mostly the DNA is present in a region, but it is not surrounded by any membrane. So there is no defined nucleus present. The region where the genetic material is present in prokaryotes is called nucleoid. Okay. So it is called nucleoid. So it is the region where the genetic material is present, but there's no membrane present around it. But in eukaryotes, there's a defined membrane present, and inside that membranous structure, the DNA is present. Okay, so it has a nucleus, proper nucleus. This is the major difference between prokaryote and eukaryote. Then comes the DNA. So in prokaryotes, the DNA is single cell circular chromosome and it lacks histones. So histones are the proteins that around which the DNA is wrapped in case of eukaryotic cells. So eukaryotic cells have multiple linear chromosomes with histones. So basically DNA for eukaryote, since it's a larger cell, the DNA is also longer in length, and that DNA is very highly organized in the eukaryotic cell. So they are folded and wrapped around proteins called histones, and then they are organized in special structures called chromosomes. So they are basically wrapped and turned and bound in a very organized manner. So they have multiple linear chromosomes with histones. So chromosomes have these histones. Okay. So and the number of chromosomes can differ from individual to individual. So humans have 23 pair of chromosomes, and other organisms have other numbers of chromosomes. Okay. Then another major difference between prokaryote and eukaryote is the membrane. So I told you, like, there's no membrane, internal membranes present in the prokaryote. So prokaryotic cell is a unicellular structure which can have a cell wall and a cell membrane, but inside it everything is present just in the cytoplasm. Okay. There's no compartmentalization inside. There's no internal membrane, internal structures like this with a internal membrane. So membrane enclosed organelles are not present in prokaryotes. They are present in eukaryotes. Okay. Then flagella generally consists of two protein building blocks in prokaryotes, but in eukaryotes it is complex, consists of multiple microtubules. Then glycocalyx present as a capsule or slime layer in prokaryotes, but in eukaryotes it is present in some cells that lack a cell wall. About cell wall, prokaryote usually in prokaryote cell wall is present, and they are chemically complex. Typical bacterial cell wall includes peptidoglycan. And in eukaryotes, it is not necessary that all eukaryotic cells will have the cell wall. So human cells or animal cells do not have cell wall. Plant cells do have cell wall. So present in some cells that lack a uh, cell wall. When present, they are chemically simple. Okay. And plasma membrane, there are no carbohydrates or sterols present in the plasma membrane of prokaryotes, but sterols and carbohydrates are present in the plasma membrane of eukaryotes. Okay. Then cytoplasm doesn't have any cytoskeleton or cytoplasmic streaming because there's no membrane bound organelle present. So there's no need of streaming in the cytoplasm. Everything is just floating around, swimming around in one pool. Then the ribosomes are smaller in size in prokaryotes, larger and smaller both in eukaryotes. Cell division is generally through binary fission in case of prokaryotes, but in eukaryotes, proper mitosis and meiosis happen. In prokaryotes, basically only mitosis happens, but in eukaryotes, meiosis also happens because eukaryotes are able to reproduce through sexual methods, whereas prokaryotes can only reproduce through asexual method, that is binary fission. Okay. So that was all about eukaryotes and prokaryotes. Uh, is my slide not visible to everybody, or is it your personal network problem? Okay, great. So I would request you, if it is still not visible, you can disconnect and join the call again. Anybody has any doubt in the differences between prokaryotes and eukaryotes, or in general definition of what are prokaryotes and what are eukaryotes? If you have any doubt, let me know in the chat box. I'll move on to the next question. Question number 6. Overall shape of a bacterial cell is determined by which of the following? Option A, cell wall. Option B, cytoplasm. Option C, nucleoid. Or option D, cell surface membrane. Overall shape of a bacterial cell is determined by which of the following? Cell wall, cytoplasm, nucleoid, or cell surface membrane. Shape of a bacteria. How do you determine a shape of a bacteria? It should be the cell wall or the cell cytoplasm or the nucleoid or the cell surface membrane. If you know the answer, please write it in the chat box. Only one person is giving the answer. What about others? The correct answer is cell wall. So cell wall basically is the outermost layer of the cell and it determines the shape of the cell. The cell wall determines the shape of the cell. So this is how a typical prokaryotic cell looks like. So here you have the cell wall. From the cell wall, there are certain uh, pillars or flagella it can protrude. The cell wall, then inside the cell wall, you will have the cell membrane, and then inside the cell membrane, it will be filled with the cytoplasm. So cell wall, then plasma membrane or cell membrane, and then inside the plasma membrane, you will have the cytoplasm filled. And then I told you there's a region where the DNA is present, that is the nucleoid. So imagine it like a hollow structure which is filled with a liquid. So this structure is filled with a liquid. It has a outermost covering, which is called cell wall. Then there is a thin inner covering which holds the liquid inside the cell, and that thin inner covering is called the plasma membrane or the cell membrane. And then this liquid is called the cytoplasm. And then inside this liquid, all the other necessary components of the cell are floating around. So genetic material DNA will be there. So DNA is localized inside this liquid. It is localized to one region, but it does not have any enclosed membrane around it. So that region is called the nucleoid region, where the genome is present. And the shape of any cell will be determined by the cell wall. So if the cell wall is spherical in shape, the cells will be spherical. If the cell wall is rod shaped, then the cells will become rod shaped. So the bacteria have multiple shapes. It could be round, spherical, so that are called cocci or coccus in singular, cocci as plural. Then if it is rod shaped, it is called bacillus or bacilli. Okay. Then if it is comma shaped, then it is called vibrio. So there are different names given to different shapes. Okay. Then if it is rounded rod, like it is rounded from the edge, but it is elongated also. So it is called cocobacilli. Okay. So cocci plus bacilli. So cocobacilli. Okay, so like that. And the cell wall structure, there are two types of bacteria, gram positive and gram negative. So cell wall structure is different. The major difference is in the thickness of the peptidoglycan layer. So bacterial cell wall is composed of a structure called peptidoglycan. So peptidoglycan, by the name you can see it has peptide and it has glycan. So it has alternating units of the glycan part, that is N-acetyl glucosamine and N-acetyl muramic acid, that are called NAM and NAG as short form. So NAG and NAM. So these are the alternating units of NAM, NAG are present. So like this, alternating units are present, and then to these NAM-NAG, peptides are attached. So a tetrapeptide is attached to which a pentaglycine. So total 9 amino acid peptide is attached to it. And these peptidoglycan structure, they are present as a thick layer in case of gram-positive bacteria, and a thin layer as in case of gram-negative bacteria. And gram-negative bacteria has another outer thin lipid membrane present after the cell wall. Okay, so this is all about bacterial cell wall. If anybody has any doubt, you can ask. Any doubt? Moving on to the next question now. Question number 7. What is the genome size in million bases of yeast? What is the genome size of yeast? Option A, 12.1. Option B, 4.6. Option C, 137, or option D, 97. What is the genome size of yeast? 12.1 million bases, 4.6 million bases, 137 million bases, or 97 million bases? Anybody? Just take a guess. Yeast is a unicellular fungus. So think like that and think how big a genome can be. Just for reference, I'll tell you, humans have 3.6 billion bases. So now you can think. If Smita says 4.6, anyone else? It's okay if you don't know, just take a guess. Anybody? Anyone else wants to answer? What could be the genome size of a yeast? Okay, I have another answer, 12.1. So that is the correct answer actually. Very good. The 12.1 million bases is the size of the genome of yeast. So here I have added the genome size of multiple organisms that are present. So first is humans. I told you we have 3.2 billion bases. Sorry, I said 3.6, I think. 3.2 billion bases uh, of genome is present in humans. Then in lab mouse, uh, the genome size is 2.6 billion and estimated number of gene is almost similar to humans, that is 30,000. So humans also have 30 to 40,000 genes. Then comes mustard weed or Arabidopsis thaliana. So it's a very classic uh, example of a, like, it's a very classical organism whose genome has been studied because among the plant species it has the smallest genome, so it was very easy to sequence it and study. So it is used as a model organism among plants. So it has 100 million bases and 25,000 genes. Then the roundworm, that is C. elegans, it is also used as a model organism for multiple studies. So C. elegans uh, are used to study uh, the localization of proteins and all, because it has kind of a translucent body. So it is easy to localize a protein tagged with a fluorescent tag. So C. elegans have a 97 million bases genome and 19,000 genes. Then fruit fly, Drosophila melanogaster. So it is also a very uh, common model organism. So people use Drosophila as a model organism to study developmental stages even in humans. So they have some similar genes as human developmental genes. So 13,000 genes are present and 137 million bases are there in the genome size. Then comes the yeast, that is Saccharomyces cerevisiae, that is the scientific name for yeast. And it has 12.1 million bases in its genome and approximately 6,000 genes. Then comes a bacterium, that is E. coli, Escherichia coli. It is also a very commonly used model organism and it has a 4.6 million bases of genome and 3,200 genes. Then comes viruses. So among virus, we have HIV, which has 9,700 bases genome size and only 9 genes. And then comes coronavirus, which is a very recent virus and it has a very large genome as compared to HIV, if you see. So that is all about the genome sizes. If you have any question related to this, you can ask. Let's move on to the next question otherwise. Flow of genetic material in microbial cells usually take place from option A, RNA through DNA to protein, option B, DNA through RNA to protein, option C, proteins through RNA to DNA, and option D, none of these. Flow of genetic material in microbial cells usually take place from RNA through DNA to protein, or DNA through RNA to protein, or proteins through RNA to DNA, or none of these. What do you think is the right answer? How the genetic material, genetic information flows in a microbial cell? Does it go from DNA to RNA to protein, or it goes from RNA to DNA to protein, or protein to RNA to DNA? It's Smita says RNA through DNA to protein. Okay. The genetic material is what? DNA or RNA or protein? What is the genetic material of a microbial cell? Of a bacteria? Let's talk about bacteria. Yeah, so how do you think the genetic material will flow? How do you think the genetic material will flow if the genetic material starts from DNA then? Option C, proteins through RNA to DNA. Yes, it should be DNA to RNA to protein. So that is the correct answer. So this is actually called the central dogma of biology. So the central dogma of cells. So you have the DNA as the genetic material. It goes to RNA and then it goes to protein. So the genetic material first replicates. So this process is called replication, where more copies of DNA are formed. So if a cell is dividing, it will form a duplicate copy. So there will be two copies of DNA first. Then that DNA will be transcribed into RNA. So this will be transcription. This process is called transcription, where the DNA will be transcribed to RNA, and then this RNA will be used as a template to synthesize the particular protein. So this will be translation. Okay. So this RNA will be then translated to protein. So this RNA will be used, first DNA will be used as a template to form another copy of DNA. Then that DNA will be used as a template to form RNA. Then that RNA will be used as a template to form protein. Okay. So it goes from DNA to RNA to protein, and then those proteins will perform certain functions to form the whole cell. So you will have one cell with the genetic material. So this DNA will first replicate and make two copies of DNA inside the cell. So you will have two copies of DNA. Then that DNA will form the RNA. So from the DNA, we'll have the RNA. From the RNA, we'll have the proteins, and that protein will now help in division of the cell, and that's how we'll have two cells with individual DNA that comes from the parental cell. Okay. So this is how the genetic information flows. So information is stored, processed, and replicated. So information is stored in the DNA. Genetic information in the form of DNA is copied into the daughter cells, that is replication. Each strand of DNA then serves as a template for generating RNA in the process of transcription, and then RNA in turn serves as a template for producing specific proteins in the process of translation. This is clear to everybody? How the genetic information is flowing? Anybody has any doubt in this? Please ask if you have any doubt in this. That's clear? Okay. Okay, then moving on to the next question. Property of a codon for always coding a specific amino acid is called. Property of a codon for always coding a specific amino acid is called. Option A, degenerate. Option B, non-overlapping. Option C, non-ambiguous. And option D, none of these. What is the property of a codon for always coding a specific amino acid called? Degenerate, non-overlapping, non-ambiguous, or none of these? So a genetic codon, it codes one codon codes for one amino acid only. That property is called what? What do you think would be the answer? Anybody? If anyone knows the answer, please write it in the chat box. Okay, Smita says option A. What about others? Is it degenerate? Is it non-overlapping? Or is it non-ambiguous? Or is it none of the above? Okay. So the correct answer is actually option C, non-ambiguous. So degeneracy of a codon is a different thing. I'll explain it in the next question. So we have genetic codons. Basically, we have four nucleotide bases, right? A, T, G, and C. This is the building blocks of our DNA. Okay. So this is the building block of DNA. And then different combinations of ATGC in triplets. So genetic codon is in triplet. So codons are in triplets, right? So now four codons, and now it can be in any of the combinations with three. So it could be ATG, it could be TGC, it could be GAT, CAT, anything it can be. So you have four codons and they have to be in triplets. So, and then it cannot, the pattern cannot repeat itself. So total, if you do the permutation combination, you will find 64 codons are possible. Okay. So it is basically 4 into 4 into 4 into 4. Sorry, 4 into 4 into 4, triplet, right? So in all the three places, probability is 4 bases, right? So 4 into 4 into 4. So 64 codons are possible. So out of these 64 codons, we have 3 codons that codes for stop codons. Okay. Other than that, 61 codons codes for amino acid. Okay. 61 codons are coding for amino acids. Now, how many amino acids do we have? We have only 20 amino acids. Okay. We have only 20 amino acids. So 20 amino acids are coded by 61 codons. So, but still one codon codes only for one amino acid. There can be multiple codons that are coding for the same amino acid. That is the property, that is degeneracy. But one codon codes for only one amino acid. That is called non-ambiguous nature. Okay. A single codon always codes for the same amino acid, and this describes the non-ambiguous nature of a genetic code. So these are the properties of a codon. So codon is a triplet. It is degenerate. It is non-overlapping. It doesn't overlap with any other codon. It is comma-less. There's no gaps in between. It is unambiguous. That is, one codon codes for one amino acid. And it is universal. So in humans, like if ATG is a codon and it codes for methionine, which is an amino acid, it will code for methionine in humans, in a mouse, in a bacteria, in a virus, anywhere. If ATG is present, ATG will code for methionine. Okay. So that is how it is universal in nature. Genetic code is called degenerate code because, so I've already explained, there are 64 codons for 20 amino acids. One codon has many amino acids. Each codon codes for only one amino acid, or more than one codon codes for single amino acid. I've already explained degeneracy, so you should be able to answer this. So degeneracy, I told you. What is it? One codon has many amino acids, or each codon codes for only one amino acid. More than one codon codes for single amino acid. What do you think is degeneracy? So each codon codes for single amino acid. It's not degeneracy. It's non-ambiguous. You know that from the previous question. So what do you think is the answer? And if each codon is coding for one amino acid, then one codon cannot code for many amino acids. That is the contradictory statement. One codon doesn't code for many amino acids. One codon codes for one amino acid. That is the unambiguous nature or non-ambiguous nature of codon. So this is non-ambiguous. This is the non-ambiguous property. This is wrong statement. So the correct answer is more than one codon codes for single amino acid. 64 codons also doesn't code for 20 amino acids. 61 codons code for 20 amino acids. Okay. So there are 64 possible combinations of 3-letter nucleotide sequence that can be made from 4 nucleotides. Of these 64 codons, 61 are used to code 20 amino acids. 3 are stop signals. Although each codon is specific for only one amino acid, that is the non-ambiguous property, genetic code is described as a degenerate or redundant because a single amino acid may be coded for by by more than one codon. So here you can see both UUU and UUC, both are coding for phenylalanine. Here you can see UCU, UCC, UCA and UCG, they are coding for serine. And here you can see CAU and CAC, they are coding for histidine. Okay. So if you see AAA and AAG, they are both coding for lysine. So you can see there are more than one codon coding for the same amino acid. So there's a chance that in my genome, AAA is present for lysine, and in a bacterial genome, AAG is present in lysine. So this is a degeneracy or a redundant property of genetic code. Is this clear? The difference between non-ambiguous and degeneracy. Is this clear to everybody? Anybody has any doubt in the properties of codon or understanding what degeneracy is? The degenerate property and non-ambiguous property, the difference is clear? Are you listening to me? Am I audible? Is it clear? Okay. If you stop responding to me, I would expect that maybe you can't hear me. So at least keep responding so that I won't think that there's a issue with my network. Okay. Okay. If it is clear, then we can move on to the next question. Question number 11. Studies of similarities and differences among the genomes of multiple organisms is carried out in. Option A, proteomics. Option B, functional genomics. Option C, structural genomics. And option D, comparative genomics. Studies of similarities and differences among the genomes of multiple organisms are carried out in proteomics, functional genomics, structural genomics, and comparative genomics. Read the names carefully. Think of the meaning, like the English meaning, and think what could be the answer. Okay, anyone else wants to answer? Okay. Option, I have one answer as option D. That is the correct answer, comparative genomics. So comparative genomics, by the name you can see, you are comparing two genomes. So you are studying the similarities and differences among the genomes. So comparing the genomes is comparative genomics. It is the study of the differences and similarities among the genomes of different organisms. Other than that, proteomics is basically the study of all the proteins present in an organism. Then functional genomics is the study of the dynamic expression of the gene products and in a specific context. For example, at a specific developmental stage or during a disease. So, what is the function of a particular gene at a particular developmental stage? So that is functional genomics. And then structural genomics refers to the high throughput 3D structure determination and analysis of biological macromolecules. So you are studying the structure of a particular protein that is expressed at a particular that is expressed during a particular disease. So that kind of thing. Okay, so that is structural genomics. Okay, moving on to the next question now. Which of the following may use RNA as its genome? So till now we have been studying the DNA as the genetic material, but there are certain organisms which can use RNA as its genome. So option A, bacterium, option B, archaeon, option C, virus, and option D, eukaryote. What do you think? Which of the following may use RNA as its genome? Bacteria, archaeon, virus, and eukaryote. Okay. I have one answer, option C. What about others? Anyone else wants to answer? Which of the following may use RNA as its genome? Bacteria, archaea, virus, or eukaryote? No one else wants to answer? So option C is the correct answer. So virus can have RNA as a genetic material. Uh, so you all know about coronavirus. So that is a classical example of a RNA virus. So the coronavirus had the single-stranded RNA as a genetic material. So whenever virus has RNA as the genetic material, when they infect the host, that is, in case of corona, it was human. When the coronavirus was infecting humans, what it does is it converts its RNA into DNA inside the host, inside the individual it is infecting. Okay, so inside our body, the virus was converting its RNA into DNA and then replicating and multiplying in numbers and then spreading across. Okay, so I'm giving corona as an example because that is something you all are aware of. So it would be easier to relate. Other than that, HIV also is a RNA virus, whereas hepatitis is a DNA virus. So there are viruses which have DNA and there are viruses which have RNA as a genetic material. Okay, all other organisms only have DNA as their genetic material. Okay, moving on to the next question. Question number 13. What is the type of extremophile that thrives in areas with very little water where it is really dry? What is the type of extremophile that thrives in areas with very little water or where it is really dry? Option A, halophile. Option B, thermophile. Option C, xerophile. Option D, radioresistant. What is the type of extremophile that thrives in the area with very little water where it is really dry? Halophile, thermophile, xerophile, or radioresistant? What do you think would be the answer? Anybody? Okay, I have one answer. Maybe option B. Okay. Anyone else? Come on, what about others? What type of extremophile can thrive in areas with very little water? So the correct answer is option C, xerophile. So xerophile is an extremophile. It is an extremophilic organism that can grow and reproduce in conditions with low availability of water. So in very dry areas, these xerophiles can grow. They require very little amount of water. Thermophiles are actually the organisms that can thrive in areas with high temperatures. So you know about hot water springs, right? So there are hot water springs, or we call it kunds in Hindi. So uh, in lot of places in India, we have these hot water springs. So the water is having very high temperature, and there are certain microorganisms that can actually grow in that high temperature water. So those are the thermophiles. Thermophiles basically they can tolerate high temperatures. So they are heat loving. So thermo is for the higher temperature. Phile is loving. Okay, so xerophile. So xerophytic conditions are basically the dry conditions. So zero is for the dry part and phile is the loving. Radio resistant is they are resistant to the radiations. Okay. What type of extremophile that lives in very salty environments such as Great Salt Lake? What type of extremophile lives in very salty environment? Option A, halophile, thermophile, xerophile, or radiophile. Now answer. I've already explained most of the terms. So what do you think for salty environments? What is the type of extremophile that can live in salty environments? High salinity. Yes, it is option A, halophile. So halophile, halo is the term used for high salt concentration, high salinity, and phile is again loving. So halo loving. Uh, so halophiles are a group of organisms with the ability to prosper in hypersaline conditions. In this kind of environment, extreme environment, the salt concentrations are in excess of seawater and can reach 20% to 30% of NaCl. Halophiles require high salt concentration to thrive in these unusual habitats. So in our environment, there are extreme condition. There are certain places with extreme conditions. So, and these microorganisms are present almost everywhere. So there are microorganisms that are present near the volcanic eruption areas, and there are microorganisms that are present in Arctic regions where it is uh, below freezing point. So in those harsh conditions also these microorganisms are present, and they are called extremophiles because they can grow in extreme environmental conditions. And one of these extremophiles are halophiles that can grow on very high concentration of salt. There are certain organisms, like I mentioned, can grow in Arctic regions. So they are called psychrophiles. They can tolerate very low temperatures. Okay. Then there are thermophiles, as I had mentioned earlier, which can tolerate very high temperatures. So like that, we have different types of extremophiles. Okay. Okay, let's move on to the next question. If you have any question, you can write it in the chat box and I can try to answer it. Question number 15. The binomial name of a microbe is composed of option A, its kingdom and genus names. Option B, its genus name and order name. Option C, its family and class names. And option D, its genus and species name. The binomial name of a microbe is composed of kingdom and genus name and order name, family name and class name, and genus name and species name. Okay. If Smita says option D, what about others? I'm getting answers only from one person. What about others? You don't want to answer? Okay. So both of you are agreeing to option D. That is the correct answer, genus name and species name. So this is how we describe the name of any microbe. So the nomenclature of a bacteria refers to and bacteria, naming the bacteria and other organisms. They are named according to the binomial system, which was introduced by Carl Linnaeus. This means that the bacterium has a name which is composed of a genus name that tells you to which genus it belongs, and a species epithet which together with the genus name that is unique to the bacterium. Okay. So here an example is given, Moraxella bovis. So Moraxella is the genus to which this bacteria belongs to, and bovis is the species epithet that indicates that the bacterium has been isolated from a cat cattle. So it is isolated from a bovine species. So it is, that's why its name as Moraxella bovis. Okay. The genus name and the species epithet form together the scientific name of the species, which is always written in italics. So whenever you are writing it, whenever you are typing it, you should always write the genus name with a capital letter and the species name with a small letter, and they should be italicized. If you are writing it on your notebooks using a pen and paper, then you cannot really differentiate between the italics and the non-italic part. So in that case, what we do, we generally write it and underline uh, the genus and the species name. So if I'm writing E. coli, that is Escherichia coli. So if I'm writing Escherichia coli, so I can't write in italics. So what I'll do, I'll just put an underline below the name. So whatever sentence I'm writing, if I'm writing Escherichia coli, I have to underline the name, and the genus name will always be written with a capital letter, and the species name will always be written in the small letter. Escherichia coli belongs to the genus Escherichia. So this is the genus. And since it is isolated from the colon of the, so basically from the gut or the colon, so it is called coli. The species is coli, and the species is always written with a small lowercase alphabet, and genus is always written with an uppercase or capital letter. Okay, this is clear. The nomenclature part is clear to everybody. Is it clear to everybody? Okay. Moving on to the next question. Gram-negative bacteria that causes stomach ulcers is option A, Helicobacter pylori, option B, Bacillus anthracis, option C, Clostridium botulinum, option D, Neisseria meningitis. Okay. Helicobacter pylori, Bacillus anthracis, Clostridium botulinum, or Neisseria meningitis. Gram-negative bacteria that causes stomach ulcers. Is it Helicobacter pylori? Is it Bacillus anthracis? Is it Clostridium botulinum? Or is it Neisseria meningitis? Okay. Dilip Kumar says option B. What about others? What do you think could be the causative agent for stomach ulcers? Okay. So we have B and then 2D. Neisseria meningitis. Okay. Anyone else? Anyone else wants to answer? So the correct answer is option A, Helicobacter pylori. So Helicobacter pylori is a gram-negative bacterium that causes stomach ulcers. The other options that are Bacillus anthracis, Clostridium botulinum, and Neisseria meningitis. So Bacillus anthracis causes anthrax, and it commonly affects the domestic and wild animals around the world. but people can get sick with anthrax if they come in contact with the infected animals or contaminated with animal products. So, if uh, if a person works in farm areas or they're constantly in contact with animals, and if the animal is infected with Bacillus anthracis, the person can also get sick with anthrax. Okay, so it causes anthrax. Then comes Clostridium botulinum. It is a bacteria that produces a dangerous toxin called botulinum toxin under low oxygen conditions. And this toxin, uh, these toxins are one of the most lethal substances known, and these toxins block the nerve function, can lead to respiratory and muscular paralysis. So, you must have heard about Botox, right? So, Botox is this botulinum toxin. So, the name comes from botulinum toxin only. And so, Botox is actually used to block these nerve functions. So, people do Botox to reduce the wrinkling of the skin, right? So, most of the celebrities, they do Botox treatment so that their skin doesn't get wrinkles. Wrinkles are basically happening because of your muscular movement, because your skin and nerves are functional there. If you block the nerve function on the skin, and so your muscles will undergo sort uh, your muscles will go through a state of paralysis. So now your muscles won't move. So your muscles won't be able to grow further or shift further. So they will be tight and they won't form the wrinkles in the face. So that is how these toxins, these are actually very lethal, but they are, and in very small quantity when used, they can be used as a cosmetic procedure where these can be used to prevent aging by preventing the wrinkle formations on the face or on the skin. Okay, so this is important uh, organism in the cosmetic industry. Okay, then third is Neisseria meningitidis. So you must have heard about meningitis or meningococcal disease. So basically, it is also in common language, it is known as brain fever. So it basically affects the central nervous system and it causes the meningococcal disease. Okay. So these were the three options. The fourth option was Helicobacter pylori, that was actually responsible for causing the stomach ulcers. This is clear to everybody? Okay, moving on to the next question. Who isolated who isolated Streptococcus pneumonia for the first time? Option A, Robert Koch. Option B, Edward Jenner. Option C, Anton von Leeuwenhoek. And option D, Louis Pasteur and George Sternberg. Who isolated Streptococcus pneumonia for the first time? I have one answer, option C. What about others? Anyone else wants to answer? Who isolated Streptococcus pneumonia for the first time? Robert Koch, Edward Jenner, Anthony von Leeuwenhoek, or Louis Pasteur and George Sternberg? Anyone else wants to answer? So the correct answer is Louis Pasteur and George Sternberg. So they both isolated the Streptococcus pneumonia simultaneously, but they did not isolate it together. Interestingly, they both were working in different places, but at the same time, they both isolated in their own labs this causative agent of pneumonia, uh, that is Streptococcus pneumonia. I told you about Robert Koch and Edward Jenner. Edward Jenner was the one who discovered the first vaccine for smallpox. Then Robert Koch gave the Koch postulate, gave the germ theory of disease. Leeuwenhoek, I told you, that is known as the father of microbiology. He was the first to observe a bacteria and a parasite under the microscope. Okay, moving on. Question number 18. A newly discovered microscopic structure is hypothesized to be a living organism. Which of the following lines of evidence would support the contention that this organism may be alive? Option A, it contains DNA. Option B, it is made of single cell. Option C, it utilizes energy. And option D, all of these. The newly discovered microscopic structures hypothesized to be a living organism. Which of the following lines of evidence would support the contention that this organism may be alive? So I told you about the characteristics of a living organism, remember? So it had multiple points in that. Think of that and tell me if you see a structure and if what properties should it have to be considered as living organism. So should it contain DNA? Should it be made of single cell, or it should utilize energy, or all of these should be present to be considered as living organism? How much time left for this? Dilip, the session will go until 9 p.m. So around 15 to 20 minutes. Okay, if Smita answered option A, it contains DNA. Okay, anyone else wants to answer? Anybody else knows the answer to this? Should that be the only criteria? It contains DNA. Don't you think a living cell should be able to utilize energy? So the correct answer actually is all of these. It should be able to utilize the energy and it should contain a DNA. And since it's a microscopic structure, it should be made of a single cell. Okay. So made of a single cell is not a property or characteristic of a living organism, but containing a DNA and utilization of energy is the characteristic of the living organism. So these were the characteristics of living organism. I've already gone through this one. So I've just added it for your reference. You can go through it later. Okay. Is that clear now? Moving on to the next question. The virus is considered to be non-living when it is outside the host because option A, it cannot reproduce. Option B, it cannot utilize energy. Option C, it is mobile. And option D, it is made of single cell. Virus is considered to be non-living when it is outside the host because it cannot reproduce, or it cannot utilize energy, or it is mobile, or it is made of a single cell. Smita says option B. Okay. Anybody else wants to answer? Okay, if I tell you that This question might have two correct answers then. Do you want to attempt this? Option B is one of the correct answers. What do you think is the what do you think is the second correct option? Yes, A and B. That is absolutely correct. So it cannot reproduce on its own. Okay, a virus outside the host cell is considered non-living because it cannot reproduce on its own. Inside the host cell, it uses the host machinery. It uses the host proteins and cellular uh, biomolecules that and reproduce and multiply inside the host. But outside the host, it is as good as a non-living object. It does not have any uh, capability of reproducing or multiplying, and hence it cannot utilize any energy. It is mobile. Saying that it is mobile is partially correct, partially wrong, because since it is non-living outside the host, so mobility doesn't really affect. So it is not really mobile outside the host, and then it is made of a single cell. That is true, but that is not a defining characteristic of a living or non-living object. Okay. So virus is considered to be non-living when it is outside the host because it cannot reproduce and utilize energy. Is that clear? Is that clear to others? Okay, moving on to the next question. What is Whittaker's classification based on? Option A, mode of nutrition. Option B, cellular organization. Option C, presence or absence of nucleus. And option D, all of the above. Yes, do you have any question? Somebody is raising hand. Do you have a question? Abhigumma. Do you have a question? What is Whittaker's classification based on? Mode of nutrition, cellular organization, presence or absence of nucleus, or all of these? What do you think is the right answer? Cellular organization. Okay. Anyone else wants to answer? So the correct answer is actually all of these. The Whittaker's mode of classification was based on multiple characteristics. So it was based on mode of nutrition and then cellular organization, presence and absence of nucleus. So uh, the answer is all of these. So this is how kingdom classification was done by Whittaker. So he divided all the organisms into 5 kingdoms. It was Monera, Protista, Fungi, Plantae, and Animalia. So Monera had all the prokaryotes. That means it had all the bacteria and archaea. So prokaryotes, I told you prokaryotes and eukaryotes had certain differences. We had already discussed the difference. So all the other kingdoms, Protista, Fungi, Plantae, and Animalia, they are under the eukaryotic part. Okay. So prokaryotes are, I told you, they are unicellular. So cellular organization, based on cellular organization, the division is clear. Then nucleus, I told you prokaryote don't have a nucleus, and eukaryotes have. Now, among these eukaryotes, you can see I told you eukaryotes can be unicellular or multicellular. So they have divided the unicellular eukaryotes into protista and fungi. So fungi can have both unicellular and multicellular. Protistas are all the protista are unicellular organisms. And plantae and animalia, they are higher level organisms. So they have tissues and organs, not just uh, cellular organization. They have a tissue organization or organ system organization. So in animals, we have tissues, they form the organs, and then we have the organ system. Like we have a digestive system which have multiple organs. It has stomach, it has intestine. So those are organs. They all together form a digestive system. Okay. So then we have respiratory system, circulatory system, all kinds of organ systems we have. So these are the 5 kingdoms that were defined by Whitaker. You can go through all of the properties. Mode of nutrition, whether they are taking the nutrition uh, from sunlight, whether they are taking the nutrition from some chemical, whether they are taking the carbon from the CO2 as carbon source or from organic material. So based on that, different mode of nutrition is present in different kingdoms. Okay. This is clear, everybody. Whitaker's classification. That's very important topic. Is it clear to everyone? Moving on to the next question then. Photoheterotroph. Option A, get energy from light and feed on other organisms. Option B, get energy from light and are self-feeding. Option C, derive energy from chemicals and are self-feeding. And option D, derive energy from chemicals and feed on other organisms. Photoheterotrophs are Ipsnita says option A, get energy from light and feed on other organisms. That is the correct answer. So from the name itself, you can identify if the name has photo, that means they are getting the energy from light. So I have this. So you can see all the organisms, they are either taking the carbon from CO2 or from organic carbon. Okay. Carbon sources either inorganic CO2 or organic carbon. So if it is inorganic CO2, they are autotroph. If it is organic carbon, they are heterotroph. Okay. Now comes the energy. So if they are taking the energy from light, they will be photoautotrophs or photoheterotrophs. If they are taking the chemical energy, it will be chemoautotrophs or chemoheterotrophs. Okay. So in the question we had photoheterotrophs. So they were getting the energy from light and feeding on other organisms, feeding on organic carbon, right? So organic carbon is basically all the other organisms or like any form of organism. If a animal is feeding on grass, that is also an organism, right? It also comes under the Whitaker's 5 kingdom classification. So in that sense, you have to think. And then whether it is using H2O, photoautotrophs, whether they are using H2O to reduce this carbon dioxide. If they are using the H2O, then they are oxygenic photosynthesis, like plant is an example. And if they are not using H2O to reduce the CO2, then it is anoxygenic photosynthetic bacteria, like green and purple bacteria. They generally use H2S instead of H2O. Okay. Then under heterotrophs, you have photoheterotrophs, you have green and purple non-sulfur bacteria. And then under chemoheterotrophs, you have, if the terminal electron acceptor is oxygen, then in that case, it will be aerobic respiration. If it is not oxygen, then it either could be organic compound. In that case, it would be fermentation. If it is inorganic compound, then it is anaerobic respiration. Okay, so there are different modes of uh, nutrition, you can say. So based on that, there's a whole classification. So you can go through this. If you have any doubt in this, please ask. Anybody has any doubt in this? This is clear to everybody? Okay. Which of the following nitrogenous bases is a purine? Which of the following nitrogenous base is a purine? Option A, adenine, option B, thymine, option C, cytosine, and option D, uracil. So these are the nitrogenous bases that makes up the DNA. So which of the following is a purine? So there are two types of nitrogenous bases, purine and pyrimidine. So which of the following is purine? Adenine, thymine, cytosine, and uracil. Okay, we have two answers. Option D, that is uracil. Okay. Surprisingly, it is not the correct answer. The correct answer is adenine. So you have two types of nitrogenous bases, pyrimidine and purine. So the name comes from the nitrogen, uh, the basic structure of the base, these bases. So you see the cyclic structure that is present in cytosine, thymine, and uracil. It is the pyrimidine structure, and in case of adenine and guanine, it is the purine structure, which is a double ring structure. So cytosine, thymine, and uracil comes under the pyrimidine bases, and adenine and guanine comes under the purine bases. Okay, is this clear to everybody? Is that clear? Okay. Moving on to the next question. Which of the following differentiates a plant cell from an animal cell? Which of the following differentiates a plant cell from an animal cell? Option A, presence of ribosome. Option B, presence of cell wall. Option C, presence of a nucleus. And option D, none of the above. What do you think? Difference between a plant cell and an animal cell? Presence of ribosome, presence of cell wall, presence of nucleus, or none of the above? Yeah. So three people are saying option B, presence of cell wall. That is the correct answer. So plant cells have a cell wall, but animal cells don't. Plant cells also have a chloroplast, which is absent in animals. And other than that, all the other structures that are mentioned, ribosome and nucleus, that is present in both plant and animal cells. So nucleus is present in all eukaryotes. So both plant and animal cells come under the eukaryotic uh, class. So they both have nucleus present. And ribosomes are also present in both of them. Ribosomes are the structure that are responsible for the protein synthesis. So with this, we come to the end of the session. Anybody has any doubt in any of the questions or any of the topics in this week's session? So this week mostly has theoretical concepts. So there are things that you have to remember, like there's no explanation in those things. You have to learn it and you have to remember it. So if there's no question, then thank you all for joining. I'll end this session now. See you on the next session. Thank you so much. Bye bye.